It is my great pleasure to tell you tonight about the tale of tritons and crown-of-thorn starfish, also known as COTS. This is a collaborative project with my former colleague from AIMS, Cherie Moti. So COTS outbreaks can actually decimate a reef within months. Currently, manual culling is one of the things that the government in Australia is doing to fight to prevent COTS outbreaks. Manual culling, however, is labor intensive and very costly. And although is effective at a small location, is definitely not scalable to the entire reef. Tritons are a natural COTS predator. And in addition of being a predator, they also release a chemistry that induces a landscape of fear on COTS. And we believe that can be used to actually prevent aggregation of COTS and therefore outbreaks. So Cherie and her team have been running lots of experiments, behavioral experiments, where they look at how do the COTS behave when they are close to tritons. And my role has been to actually look at all those experiments from a data science perspective and try to extract information and conclusions using data analysis. So at AIMS, Cherie and her team have been running experiments over the last 5 years. So there is a lot of data and a lot of very interesting behavioral experiments that we want to analyze to better understand the interactions between these two species. In addition to all these experiments in the lab, we actually also conducted an experiment on the reef. On Rip Reef, which is a reef not too far from Townsville, a couple of years ago, we deployed 5 tritons within an acoustic array. And we follow the movements for over 41 days. So these are the tritons. So each triton had attached a transmitter, and we have the receivers that conform the acoustic array. And we follow the movements over, as I said, about 41 days. And the goal with this experiment was to try to understand the short-term activity of the tritons in the reef. How do they move? And how do they utilize the space, which is something that is not known. I should also say that there is a lot of unknown things about tritons. Like, for example, we don't really know the gender unless they lay eggs. And also, we don't really know how do they interact with COTS. So from that experiment on the reef, we obtained data, and these are the points that you see here on the screen. So we were able to track the movements of each of those 5 tritons over day and night. And that allowed us to understand and to estimate using statistics what we call the home range area. So the home range area is the area that tritons spend most of the time, the region that the animals require during their normal activities. So there is still a lot of things to learn and to analyze from the data from all these experiments. So we are hoping that the analysis of the data from all these behavioral experiments will allow us to better understand the interactions between tritons and COTS. And our goal is to understand how to also behave in captivity. So what are the predator-prey interactions? And we want to use tracking data from video footage to better understand that. And we are hoping that this research provides an insight to possible alternatives in the fight of COTS outbreaks in the Great Barrier Reef. And with this, I will conclude my presentation and invite my colleague, Cherie, to take the floor. Thank you.
Thank you very much, Patricia. I'm a natural products chemist working at AIMS, and my current area of research is the crown-of-thorn starfish, which, as we speak, are actually in outbreak status. Their main food is hard coral, and their current high populations pretty much represent a death sentence for many reefs. But before we get too down on COTS, we need to remember that this starfish is actually a native to the reef, and therefore it's important to find sustainable and safe ways to control these animals and return their populations to normal levels, and ultimately bring the ecosystem back into balance. So at AIMS, we're looking for ways to change their behaviour naturally. And so where do we start with this? Well, we know a lot about the COTS biology. We know that they have eyes and tentacles at the end of each arm. The tentacles are capable of detecting chemicals in the water that can help them find coral food, find a mate, or evade a predator such as the giant Triton snail. And this is equivalent to our sense of smell. So we are identifying what odour chemicals are released by COTS or corals or predators, and whether they can then be used as a chemical weapon against COTS. For example, if we could identify the food odour, then we could possibly identify develop a baited trap. Think about it like a mouse trap. If we could identify the predator odour, we could develop a barrier a bit like a mozzie repellent. So how do we actually do this? Well, we expose COTS to odour chemicals in controlled aquarium experiments, and Patricia has already shown the response of COTS to the giant Triton odour. What we do here is we actually put them into a system where they can get the sense of the odour, but then have the time to respond. And they either have a trajectory of fleeing or they have a trajectory of meandering. And we use what we call a Y-maze assay to actually record the information and enable us to do some statistical analysis and some modelling to see where exactly the animal goes. And we see that in this instance here, the animal that's been exposed to the giant Triton snail, that's the one on the left, actually responded quite erratically, moved completely, and then moved actually into the safety of the second arm. So using these kinds of techniques, we're actually now we're now being able to test candidates, and hopefully we'll have something in hand that we can deploy in the field in the next year or so. But I was also asked as a research chemist to talk about one of the other diverse projects that I work on. And I've been asked really to talk about my second job, which is emerging pollutants on the Great Barrier Reef. And my focus within this space is looking at plastics. We all use plastics every day, but what we don't consider is that they can break into many pieces. These pieces are so small that they can't be seen with the naked eye. So we call these microplastics. These microplastics are increasing in numbers in our oceans, and because they're microscopic, they're very, very difficult to remove. Microplastics have been found in numerous different animal species, from turtles to fish and even sea cucumbers. And we're only now beginning to investigate the impacts of these microplastics on animal health and behaviour. However, what is of most concern is the fact that the lifetime of a plastic is actually hundreds to thousands of years. So their impact will be felt now and for centuries to come. So we're actually interested in trying to quantify the microplastic pollution on the GBR. We want to know where they are, where they're concentrated, and what types they are. To do this, we need to sample the seawater using tow nets. We count and size sort the contents of these nets using sieves to recover the items that might be microplastics. We then use microscopy and chemical techniques to confirm whether an item is a microplastic. We've now conducted surface tows in numerous areas, but I'm going to show you just the section of the Great Barrier Reef closest to our hometown here in Townsville. And we were actually surprised to find that microplastics were higher in concentration on the outer reefs than on the inner reefs. And we can see on the map here that the outer reefs have got the red dot indicating higher concentration. So bring in the mathematicians again. Modelling was done to determine whether these microplastic items could possibly be coming from our rivers. And what we found is that there was actually no transport possible, no possible pathway of transport of the microplastics from our rivers to the outer reefs. So this actually suggests that the source of these microplastics is not from our shores. And when we did some potential pathway modelling, what we found is that the microplastics that were on the outer reef actually stayed and accumulated in those areas. So this is yet another area of concern. We have also concurrently undertaken a number of studies examining the gut contents of reef fish, including what I'm showing here is the small damselfish and coral trout. And we found that these species both contained microplastics. So unfortunately, what we're finding is that what is in the water is also in the fish. We are now in the process of conducting experiments to determine whether the health of these fish is actually impacted. The ubiquitous nature of microplastics is concerning, and we must all find ways to reduce the entry of plastics and microplastics into our oceans. So I hope I've given you a bit of a flavour of the work of a chemist in the marine biology sphere.